Hi everyone, my name is Anoy, and today I'm going to talk about neural tube formation. So neural tube formation starts about 3 weeks into development. And so if we look at a cross-section of the embryo, just want to draw out here. So we have the embryo there, and we have a layer of cells that forms the hypoblast right here, and underneath it we have the yolk sac, and then above we have a layer of cells that forms the epiblast, and over here we have the amniotic cavity. So what's going to happen is that the layer of epiblast cells here, I'm going to label these as well, so epiblast and hypoblast. So the layer of cells there are going to form what is the neural plate. So if we zoom in on that, so we create a new, so we're going to zoom in on this guy. We're going to get a sheet of cells like this. And these cells are going to be bound together by an adhesion belt, which is a long belt of actin filaments that connect all of these cells together. Just like that. And what we're going to see is that the contraction of this adhesion belt is going to create an entire system-like movement that is going to simulate the formation and folding of our neural tube. So from the neural plate, we are going to eventually get the neural tube, and how this happens is that the neural plate will invaginate to create a structure that looks something like this. And this happens from the contraction of this adhesion belt, and what that does for each individual cell will eventually create that belt. So we can consider one individual cell right here, and we have the actin filament across here. So to remember from cell biology, the contraction of actin occurs when you have the actin filament here and the myosin that binds, which is now, this is where the myosin is, and the myosin head protein will attach and bend further and pull this bottom sheet along. And so what will happen to this cell is that this entire area right here, this region, will contract and become a little bit smaller. And the problem is, or what happens, not the problem, but the distribution of the mass remains the same for the top and the bottom, but the area remains the same, but the dimensions change as the bottom segment of that rectangle has gotten much smaller, right? So what happens is the cell is going to flare out and look a bit of like a bottle. The reason why this is significant is if we consider one cell in the neural plate and we consider that its area got a little bit smaller, so let's make it a little bit smaller, it's going to squeeze and flare out like this. And the reason why that's going to create a conformational change in the plate itself is that its neighbor will also do that, and these two cells are going to rub against each other and push out. It's going to flare in this way because there's this tension between the two cells at that midpoint. And we can see this here if I erase these cells right here. We can see with one of these cells squeeze a little bit, I'm doing blue. One of these cells will squeeze like this, and then we'll get its neighbor here flaring out a little bit, and another neighbor on that side. And these cells are going to start clumping in this way and flare upwards like that because these cells are widening at the bottom, forcing them all to be up. And eventually what we're going to get, I scroll down, something that looks like this. with the cells now having been invaginated, forming the precursor to the neural tube. And what's going to help us finally form our eventual product, the neural tube, which will look something like this with these cells, are proteins that are called cadherins. And cadherins bind to each other and create a bridge essentially between two cells. And there are different types of cadherins. One type, which I'm going to do here in yellow, is a E-cadherin, which stands for epithelial cadherins. And these are cadherins expressed by epithelial cells. Another example, which I can do in red, is an N-cadherin, which is expressed by neurons. At this point, our cells are going to be differentiating and expressing these different cadherins based on what cell they're going to be. So what we're going to get is on the outside, we're going to get these epithelial cells start expressing these E-cadherins. And on the inside, we're going to get these neuron cells expressing these N-cadherins throughout this invaginated neural plate. And what's going to happen is that it's going to pinch off when these cadherins bind with each other and form this right here. And above, what we're going to get is the remnant of the neural tube. And above, what we're going to get is an epithelial sheet, which is connected by those E-cadherins that we saw here. So thanks for listening. Hope you learned something about neural tube formation. Thanks to Nelson Gomez for letting me borrow his equipment. Appreciate it a lot. And hope you learned something. Thanks. Okay, I think it's fine.
That sounds pretty good.
Sweet, dude. Yeah, I think just